You know, when we talk about AI in drug discovery, the popular image is this sleek, futuristic vending machine. Like you type in cure cancer, press enter, and the machine just spits out a perfect pill.
Right, yeah. The whole magic box theory. It's a really lovely fantasy.
Exactly. But when you actually peel back the layers of how computational biology has worked for the last decade or so, it's not a magic box at all. It's more like a Frankenstein monster stitched together from spare parts.
That is painfully accurate. I mean, it's a total patchwork. You have one AI model that tries to guess where a drug might stick to a protein.
Yeah.
That's docking.
Right.
Then you have a totally different model trying to design the shape of the drug from scratch, the de novo design, and maybe a third one trying to tweak it so it actually, dissolves in water.
And the problem, as I understand it, is that these models don't really
Did to speak the same language.
Right. So you're losing data at every handoff. It's totally fragmented.
Precisely. Which is why the paper we are diving into today is such a massive deal. It honestly feels like someone finally decided to stop patching the holes in the hull and just build a brand new ship.
I agree. So welcome, everyone. Today we are looking at a breakthrough paper published in the journal Cell, specifically the 2026 study by Peng and colleagues, and they built a system called PocketXMol.
And they aren't calling this just another tool either. They are pitching it as a unified model, basically a grand unified theory for molecular generation.
That's a huge claim.
It is. They claim to have collapsed all those fragmented steps into a single, seamless brain. Docking, design, optimization, all of it.
So our mission today for this deep dive is to really pressure test that claim. We need to figure out how you actually build one brain to do three very different jobs. We're going to break down this universal Denoiser concept they use pretty much sentence by sentence.
And we definitely have to look at the scoreboard. Because they didn't just stay in the simulation. They went into the wet lab, targeted a notoriously difficult protein called caspase 9, and they claim to have beaten it.
Let's start with the philosophy shift then. We can call this the atom-level unification. What exactly was wrong with the old way of looking at proteins?
Well, traditionally, biology loves labels. If you look at a protein, a biologist sees a string of amino acids: alanine, lysine, tryptophan. These are the building blocks.
The Lego bricks.
Exactly, the Lego bricks. So most previous AI models were trained to see these blocks.
Okay, but proteins are literally made of amino acids. Why is that a bad way for an AI to look at them?
Because it's low resolution. It's too rigid. Imagine you're trying to build a perfectly ergonomic handle for a custom tool. If you're forced to use big, square Lego bricks, you can only get the shape mostly right.
You can't get the subtle curves?
Right, you miss the exact friction points, the micro interactions.
PocketXMol does it differently.
PocketXMol completely ignores the bricks. It ignores the labels alanine or lysine entirely. It zooms in and looks strictly at the studs on the plastic bricks. It sees the raw atoms: carbon, nitrogen, oxygen, sulfur.
So it basically creates a cloud of geometry.
Yes, exactly. It abstracts the drug and the protein pocket, which they denote in the paper as K, into the exact same language: raw atomic coordinates.
So to PocketXMol, a protein isn't a biological sequence at all.
No, it's just a specific arrangement of physics in 3D space.
I see the advantage there. If everything is just atoms, you don't need separate rules for a small molecule versus a peptide versus a giant protein.
That's the critical takeaway here: transferability. Because it speaks the universal language of physics, it can switch between designing a small aspirin-sized drug and a long floppy peptide chain without missing a beat. To the model, they are just different arrangements of the exact same stuff.
Okay, but here's where I get stuck on the actual mechanics of it. If it's just one big brain looking at a cloud of atoms, how do you tell it what you want it to do?
Right. How does it know the task?
Yeah, how does it know if I want it to find a structure or invent a totally new one?
That is the really clever part of the architecture. They developed what they call atomic prompts, denoted as P in their equations. Think of it like a massive switchboard on an audio mixing deck.
A switchboard. Walk me through that. How does it work atom by atom?
It utilizes a binary code, zeros and ones. These are indicator variables that control the task. Let's say you have a protein pocket, and you want to invent a drug to fit inside it from scratch.
De novo design.
Right. You flip the switch for generate atom types to on, which is a 1, and you flip generate coordinates to on, another 1. You are essentially telling the model, I don't know what atoms go here, and I don't know where they go. You figure it out.
Okay, that makes sense. But what if I already have the drug? Like, I just want to see how it fits.
That's the docking task. For that, you flip the generate type switch to off, a 0, because you already know it's a carbon, a nitrogen, and so on. And you only leave the generate coordinates switch on.
So the brain itself stays exactly the same. You're just constraining which parts of the problem it's allowed to solve using this binary mask.
Precisely. It unifies structure prediction and de novo design just by toggling those inputs.
Mm.
But the engine that actually does the heavy lifting and solves it, that's the second pillar, the universal denoiser.
Universal denoiser sounds like some kind of weapon from Star Trek. What is it actually doing in the algorithm?
So in modern generative AI, we often view creation as a process of cleaning up noise. Imagine a TV screen showing pure static, black and white snow.
Right. High entropy, just visual chaos.
Exactly. The model's job is to look at that static and slowly, step by step, smooth it out until a clear image appears. But in this case, it's not an image of a face or a cat. It's a 3D structure of a molecule.
So it starts with a scrambled mess of atoms and literally organizes them.
Yes, but here is the breakthrough. Distinct tasks usually have totally different flavors of static.
What do you mean by flavors of static?
The noise you get when you're unsure about a chemical bond type is mathematically very different from the noise when you're unsure about a 3D coordinate position. Usually, you'd need a completely different AI trained for each specific type of noise distribution.
Oh, I get it. It's like trying to use a standard text spell checker to fix a blurry photograph.
Great analogy. It just wouldn't work. But PocketXMol maps all these different uncertainties into a unified noise space. It acts as a geometric neural network that can clean up any type of atomic static.
And it does this automatically.
Yes, it self-calibrates. You don't even have to tell it how noisy the data is. It identifies the noise characteristics directly from the input.
That is fascinating. Now, there's one more piece of the architecture I want to hit before we look at the actual lab results. The confidence score.
Right, the third pillar. This is crucial because AI, even the absolute best AI, hallucinates. It will confidently hand you a molecule that looks amazing on a screen, but completely violates the basic laws of physics.
The confident idiot problem.
It really is. But PocketXMol grades its own homework and uses a graph neural network, where the atoms are nodes and the bonds between them are edges, to learn latent features.
So it's mapping their relationships.
Exactly. Then, for every single atom and bond it places, the denoiser outputs confidence score, ranging from 0 to 1. It's basically saying, I'm 99% sure this carbon belongs right here, or I'm only 20% sure about this double bond.
Which allows the researchers to just filter out the garbage immediately.
Precisely. If the confidence is low, you discard it. You don't waste months and millions of dollars synthesizing a hallucination in the lab.
Okay, we are about 5 minutes in, and that was a lot of dense theory. Let's pause and anchor this with a quick recap for you listening.
Good idea. Let's break it down.
All right, here are the bullet points on the unified approach so far. Point 1, drop the labels. PocketXMol doesn't look at amino acids. It looks purely at raw atomic coordinates. Point 2, the switchboard. It uses atomic prompts, those binary zeros and ones, to tell the model whether to fix atoms in place or generate new ones.
Point 3 is the universal denoiser. It treats every problem, whether it's design, docking, or optimization, as just cleaning up static to reveal a clear 3D structure.
And point 4, self-grading. It generates confidence scores so we actually know which molecules are likely real and which are just AI hallucinations.
That's a perfect summary.
Mm-hmm.
And remember the golden rule here. It does all of this without needing task-specific fine-tuning.
So theory is great, but does it actually work? I see in the paper they ran it through a pretty intense gauntlet of benchmarks.
13 distinct computational benchmarks, to be exact.
And what was the scorecard?
It hits state-of-the-art performance on 11 out of the 13 tasks.
That is just dominant.
It really is. It outperformed 55 different baseline models. And we're talking across the board. Docking, which is putting the key in the lock. De novo design, making a totally new key.
Mm-hmm.
And importantly, peptide design.
Let's expand on that. Why is the peptide part so impressive? I feel like we glaze over that word sometimes in these discussions.
Peptides are kind of the middle children of biochemistry. They're short chains of amino acids. Too big to be traditional small molecules, but too small to be stable, folded proteins. They are floppy, they whittle constantly.
So they're hard to pin down.
Extremely. Most small molecule AIs fail entirely at peptides because they can't handle that dynamic flexibility. But PocketXMol handled both equally well because, again, it just sees atoms in space. It doesn't care about the category.
Okay, dominating benchmarks on a computer is great, but benchmarks are still just numbers on a screen. I want to talk about section 4 of our deep dive, the case study, because they took this into the real world and went after a killer.
Caspase 9.
Tell us about caspase 9. Why was this the chosen boss fight for PocketXMol?
Caspase 9 is a beast of a protein. It acts as a core regulator of cell death, specifically intrinsic apoptosis. In cancer, particularly in solid tumors, you often want to trigger cell death to kill the tumor. But caspase 9 is a major barrier in those tumor-sensing pathways.
So we want to inhibit it to help fight the cancer.
In this specific therapeutic context, yes. But the major problem is selectivity. Caspase 9 belongs to a whole family of proteins, caspase 3, caspase 7, and structurally they all look almost identical.
So it's a needle in a stack of needles problem.
That's the perfect way to put it. If you design a drug for caspase 9, it almost always accidentally hits caspase 3 as well, and that causes off-target side effects you really don't want.
So they unleashed PocketXMol on the supposedly undruggable target. What was the specific algorithm or process they used?
First, they defined the 3D coordinates of the pocket on caspase 9. Then PocketXMol generated candidate molecules. But they didn't just take the first one it spit out. They sampled 100 different candidates.
And how did they narrow that down?
They used those confidence scores we talked about, along with log-P p-values, to rank them. Log-P is essentially a measure of solubility. It tells you how well a molecule mixes with water versus fat, which is critical for a drug actually working in a human body.
And out of that ranking process emerged a winner.
Molecule 84663.
Not the catchiest name in the world, but definitely remember it.
Yeah, 84663 was a star. There was also a second-round validation molecule they called D12, but they actually synthesized 84663 in the real world, in the wet lab, and tested it against commercial pan-caspase inhibitors.
Drugs that are already out there.
Right. Established drugs like 2-VD-OFF and emercasan.
Those are heavy hitters in the oncology world.
They are, and molecule 84663 showed efficacy comparable to those established drugs. But the real smoking gun, the highest level of evidence for me, was the Western blot analysis.
Walk us through that, just for the listener who maybe hasn't looked at a Western blot since a college biology lab.
Sure. Think of a Western blot as a way to visually see specific proteins in a mashed-up cell sample. You separate the proteins by size on a gel, and they appear as dark, horizontal bands. The researchers treated MC38 cells, which are colon cancer cells, with molecule 84663.
And what did the resulting image show?
In the images, you can see very clear, dark bands for two things: cleaved PRRP and cleaved caspase 9.
Cleaved meaning chopped up, right?
Yes. This is the crucial biochemical mechanic. When caspase 9 is active and apoptosis, or cell death, is happening, specific downstream proteins like PRP get physically chopped in half. They get cleaved.
So seeing those cleaved bands, is visual, undeniable proof that the AI-designed drug successfully engaged the target and triggered the cell death machinery inside actual living cancer cells.
That is just wild to think about. We went from binary code and a prompt to dead cancer cells in a petri dish.
And it actually goes even further. They detail in vivo evidence as well, actual live subjects.
Really?
Yes, they use the peptide design capabilities we mentioned earlier to make peptides that specifically target PD-L1, which is another major cancer target. The live imaging showed these peptides specifically accumulating right inside lung tumors in the animal models.
So it's not just floating randomly around the bloodstream, it's homing in on the tumor.
Exactly. High affinity, targeted delivery.
I want to zoom in on why molecule 84663 worked when so many human-designed drugs might fail. In the paper, there's a graphical abstract that actually shows the chemical bond.
It's a really beautiful image. I highly recommend checking the show notes for this so you can visualize it. You see the drug, which is this green structure, nestled perfectly into the gray spheres that represent the protein pocket. It looks exactly like a puzzle piece clicking into place.
It does, but you have to look closer at the colors. You see specific chemical groups on the drug, blue nitrogens, red oxygens, and they are aligning perfectly with the corresponding atoms of the pocket. This is the real aha moment of the paper.
Because PocketXMol didn't just find a generic ship that fits into the hole.
Right. It optimized the specific atomic interactions. It knew that this blue nitrogen needs to be exactly right here to form a hydrogen bond with that specific part of the pocket.
Whereas a coarser model, one using those Lego blocks we talked about earlier, might have just put a generic blob of mass there.
Exactly. And a minor atomic alteration, like moving a carbon atom just 1 angstrom to the left, and an angstrom is 1/10,000,000th of a meter, that tiny shift could completely ruin the binding. PocketXMol's atom-level resolution prevents that from happening.
And they also showed it could optimize specific properties after the fact, right? This is the steering concept they mentioned.
Yes, the steering algorithm. They did a specific optimization task for log-P. that solubility metric we discussed. Log P determines if a drug can actually get through your stomach lining and into your blood.
So how do you steer an AI to fix that?
They took a starting molecule that didn't have great solubility, and they deliberately added noise to it.
Wait, they made the design worse on purpose?
Yes. Think of it like softening a piece of clay that's already dried a bit. They added noise, specifically a noise scale of 0.3, to partially scramble the atomic coordinates. Then they ran 30 denoising steps, but they algorithmically steered the denoiser during those steps.
By telling it what to aim for.
Right. They essentially told the model, clean this static up, but as you do, aim for a higher log P value.
So they didn't have to start from scratch. They just tweaked the existing working design to make it more viable as a pill.
Exactly. It successfully shifted the molecule's chemical properties while preserving the core structure that binds to the target protein.
Okay, this is a perfect place for our second recap. We're about 12 minutes in now. Let's summarize the case study and these mechanics for you guys listening.
Let's do it. Point 1, PocketXMol tackled the selectivity nightmare of caspase 9, a major cancer target.
Point 2, it produced molecule 84663, which matched established commercial drugs in sheer potency.
Point 3, the Western blots confirmed it works inside living MC38 cells, creating cleaved PIRP, which is the undeniable signature of targeted cell death.
And point 4, the model demonstrated it can optimize specific drug properties like log P by adding noise at a scale of 0.3 and refining it with 30 steered steps.
That covers the hard evidence.
I actually have a question for you listening right now. If you could design a molecule to block any protein in your body, maybe a protein that causes Alzheimer's, or one that accelerates aging, which one would you choose? Because PocketXMol suggests we might very soon have the tools to actually build it, even if we've never successfully drugged it before.
That's the ultimate dream of this field, and I think that leads us perfectly into Section 6 of our outline, the broader implications.
We touched on the peptides briefly earlier, but I want to circle back to the validation data there. How do we know the model didn't just memorize peptides it had seen in its training data?
That is a critical scientific control. When they tested the peptide design capabilities, they explicitly excluded any entries with over 30% sequence identity from the training set.
30% seems like a fairly low threshold.
It's actually a very strict threshold in computational biology. It means the model absolutely couldn't rely on memory. It had to genuinely figure out the raw physics of completely new, unseen peptide chains.
And how did it score?
It still achieved exceptionally high DocQ scores. DocQ is the standard metric for assessing quality in peptide docking. It proved it was learning physics, not just memorizing patterns.
So, synthesizing all of this, the biggest takeaway for the AI field as a whole here is the universal aspect of the model.
Absolutely. The fact that this model does not require task-specific fine-tuning, that is the holy grail of artificial intelligence, generalization. It strongly suggests a future where one central brain handles all drug discovery tasks.
I want to try one last visual analogy for the listeners. I love the Swiss Army knife image for this.
Go for it. It fits perfectly.
Before PocketXMol, doing computational drug discovery was like carrying around a heavy bag of loose, mismatched tools. You had a screwdriver for your docking model, a hammer for your de novo design model, and maybe a wrench for your optimization model, and none of them fit together well.
Right, and half the time you lost the wrench or the screwdriver stripped the screw because the data transfer between them was so lossy.
Exactly. PocketXMol is the Swiss Army knife. It is one single unified tool. You just flip out the little blade you need for the moment, which is the atomic prompt, and it does the job using the exact same handle. One tool, many distinct atomic functions.
And that handle, the core of the tool, is a deep, fundamental understanding of atomic interactions. Once the AI understands the atoms, the actual physics of the universe, you can build practically anything.
So bringing it all home, we move from binary code, literally zeros and ones in a task prompt, to real-world cancer-fighting drugs in a single unified workflow.
It's a total validation of the bottom-up approach. Don't start with the clunky biology labels. Start with the raw physics of the atoms.
Here is my provocative thought to leave you with today. If PocketXMol can design highly selective drugs for difficult targets like caspase 9 today, what happens when we apply it to proteins we don't even have 3D structures for yet?
That is the next great frontier. AlphaFold gave us the ability to predict the structures. PocketXMol gives us the ability to design the drugs that fit them. Are we entering the era of truly on-demand medicine, where if a novel virus appears or we sequence a new cancer driver in a patient, we just feed the atoms into the universal denoiser and essentially print the cure?
That is absolutely a future I want to live in. Listeners, what stands out to you from all of this? Is it the universal denoiser concept, that philosophical idea of resolving pure order from static chaos? Or is it the hard evidence that an AI designed a working cancer drug that held its own against commercial inhibitors in a wet lab?
I'm honestly still stuck on those Western blots. Seeing those dark bands appear on the gel, that is reality staring you right in the face. It's not just theoretical code anymore, it's chemistry.
Go check out the show notes. We've put the structure of molecule 84663 in there. Look at the graphical abstract, look closely, and see the individual atoms locking together.
It's definitely worth a look. It's just beautiful science.
Thanks for diving deep with us today. Keep analyzing and keep learning.
See you next time.